with, I asked you to tell me the type of diagram that's created based on the molecular differences of similar organisms when we talk about phylogeny and things of that nature. Let's see what you all came up with. Yay, a cladogram. Perfect, right off the bat. A squirrel diagram, a tree, okay. Close with a phylogenetic tree. Actually, a cladogram is a type of phylogenetic tree based on the molecular differences of similar organisms. Yes? Well, a cladogram, and I should have been even a little more specific, actually based on the definition I gave you, both phylogenetic tree and cladogram would be acceptable, but I was thinking about cladogram in my head. Cladogram, please quit talking for a minute, is based on what are called outgroups. Okay? So as soon as I don't have something in common with something else, or if I have a new trait, I become a new clade. And so it's like if I don't have this, I'm now my own group. Okay? A phylogenetic tree is just a general schematic of how things are related. All right? And we'll get into that today a little bit more. So I guess today I would have accepted either cladogram or phylogenetic. Either one is really correct based on what I said. So systematics is really what we're talking about when we start to mention cladograms and phylogenetic trees and phylogeny in general, relatedness. So it's the study of the diversity of organisms using information from cellular to population levels. Now, back in Charles Darwin's time, they did not have molecular evidence. So they had to look at traits, right, physical phenotypes, and try to determine relatedness based on what's called morphological structures. That's very difficult to do. Now, one goal of systematics is to determine phylogeny, again, relatedness, evolutionary history of species, so to speak, of a particular group. Now, phylogeny is often represented as what is called a phylogenetic tree, and a diagram indicating lines of descent is what we're talking about here. And I often, even in my own head, I really have to think specifically when I try to define a phylogenetic tree and not accidentally define a cladogram. I just did it with you. So a diagram indicating those lines of descent is what we're going to be creating and looking at, and each time you have a branching point, you're looking at divergence. So is a divergence from a common ancestor and represents an organism that gives rise to two or more new groups. We have to at least have two, otherwise you would not have a divergence, right? You'd just have a single line. And then you'd have like what we are, Homo sapiens. One single line. Unless we go back far enough, then we run into hominids and other primates. So classification lists the unique characters of each taxon and is intended to reflect phylogeny. So when we look at ancestral traits, these are present in all members of a group and present in common ancestors. So if I look at everything with a backbone, they are all, and that is an ancestral trait, the fact that we have a backbone. Everybody give me a thumbs up. We got thumbs, right? Okay. Anything with an opposable thumb is going to have a common ancestor in that case. I should say primates with opposable thumbs. And then we have derived traits. These are present in some members of a group, but absent in the common ancestors. So the fact that we walk bipedally and speak, right? Language. Sapien that's what that means, to speak, right? So we are Homo sapiens. We are the only species in our genus, Homo, because we can speak. We're a primate that speaks, walks upright, and has opposable thumbs. So that is our derived character, language. The fact that we have a hyoid bone is another derived trait. You guys know what your hyoid bone is? Let's see if anybody knows. I'll throw it out there. Don't Google it, you cheaters. What is a hyoid bone? It's the only bone in the human body that what? Come on, anatomy folks. I'm looking right at you. Oh, made me feel like I failed. It is, okay, it's white. All bones are, well, kind of. It does allow you to talk. Ah, there it is. The bone that attaches to the tongue and connects to no other bone in the body. It does not articulate with any other bone. It's the only bone that does that. It's unique to our species. It's the hyoid bone. If you just stick your finger in your mouth and keep going down until you run into the bottom of your tongue, don't do that, but you'd run into your hyoid bone. It's what your tongue attaches to, okay? It attaches to some other cartilage and some other muscle, and we control it with several muscles. But it is what gives us language and speech. It allows us to change and manipulate the size and shape of our resonance chamber. Okay, this whole vocal thing. If you actually, and I don't suggest doing it, because it hurts like all get out. If you reach back here, just underneath your mandible, okay, right, you feel these two little pokey things, and it's really sensitive. And if you were to like squeeze that, you'd crush it. That's the two tips of your hyoid bone, and it's like a U-shaped bone that your tongue, from behind, actually attaches to and allows you. And if you guys go ahead and talk a little bit and hold on to it, you will feel that it's like a little vibrating wand. It's kind of creepy, isn't it? So I'm talking right now just to feel it because it's kind of funny. But it vibrates a lot, and that's what gives you speech. Okay, and that's unique to us. So that is a good example. I'm going to break my rule. An illustrative example of an illustrative example of a derived character is indeed our hyoid bone. Okay, so we can start to classify organisms by what are called derived characters, and I'm missing part of my screen. That's not cool. Why are you doing that? Let me try to switch modes. Maybe I can. It's not helping. Well, dear, okay, we'll just look at the first one. So looking at the derived characters, in this case, we're looking at even-toed hooves being a derived character in a certain type of ruminant or mammal. Obviously, if you have mammary glands, that is a derived character for all mammals, but then based on having hooves or having tails, in this case, they're looking at the monkey's tail, and for us, we're looking at opposable thumbs here, that falls into the primate or the chimpanzee family. We would then branch off based on that derived character. Okay? So depending on your different derived characters, you'll be put into a different phylogenetic branch of the phylogenetic tree, and we'll talk more about cladograms too. So again, we can continue this till we get really specialized, and we end up with more and more derived characters, and then we start to get down into our different, in this case, families, it looks like. So looking at a better way to do it, I always say, is cladistics. Phylogeny is great. It's just too general for me. You could not prove the theory of evolution to me based on phylogeny alone, just looking at something. It doesn't work like that. Usually, looks are misleading a lot of times in nature and relatedness. So cladistics is a way to analyze primitive and derived characters and by the construction of phylogenetic trees that we call a cladogram on the basis of shared derived characteristics. They are arranged in taxa, and when you put them into a cladogram, we call them their own clades. So a cladogram is a special type of phylogenetic tree, and a clade is an evolutionary branch that includes a common ancestor together with all of its descendant species. And it traces the evolutionary history of the group being studied. And what it allows us to do is also use genetics based on the presence or absence of genes or amino acid sequences. So clades are guided by the principle of what's called parsimony. Now, parsimony I call common sense, but it means the minimum number of assumptions is the most logical. That means if I told you I have two differences in my hemoglobin protein between me and, let's say, a chimpanzee, and I have 150 between my hemoglobin and a snail, who am I more related to? The chimpanzee. It's that simple. It's saying, let's just go with the path of least resistance. Which one makes the most sense? Simplest to more complex. If I asked you which came first, multicellular or single cellular, you would tell me single cellular. That is parsimony in its simplest form. So the best cladogram is one in which the fewest number of shared derived characters are left unexplained or that minimize the number of assumed evolutionary changes. So you would want to try to find the missing link if possible, is what it's saying. We understand sometimes we don't have the missing link. So reliability of cladograms is dependent on the knowledge and skill of an investigator. When I say investigator, I'm usually talking about a geneticist when I refer to the stuff I like to look at. So if we were to not look at genetics, but let's first look at just those morphological structures, and we tried to put them into in-groups and out-groups, we would notice that we would base our relatedness on how many similar or how many different they have in common. So based on this, if I asked you, okay, well, if, and I got to get closer because I can't read right here. If I looked at these, and this is a chimpanzee, and then we go all the way down here to very early ancestors, the lanceolets, we can, and you guys probably don't even know what that is. Let's go to lizard, crocodile, and chimpanzee. We can see that a chimpanzee is not very closely related to a crocodile, but it is closer to a crocodile than it is a lizard. Okay? The only way we know that is because a lizard does not have a gizzard. That's just fun to say, right? No? Okay. And so, but a crocodile does have a gizzard. A gizzard holds like stones and things to help break down food. But a chimpanzee does not have a gizzard, but it does have like amniotic eggs, 4 limbs, vertebrae, and a notochord and embryo. Yes, indeed. Okay. So I know it sounds odd, but if you look at the genome of them, they're super similar. Okay. So I think you understand relatedness based on similarities and differences. Can I take this same idea and apply it to genetics? And that's what I would do, yes. The X means that they do have it. Okay, that's like, check, you've got it. Okay. What? Amniotic refers to the fluid or the amnion that helps develop the embryo inside. Okay. That means that the egg has the amniotic fluid within it. Okay. We're going to go to the next one. So based on what we looked at there, we can start to develop what's called a cladogram. And you'll notice a cladogram doesn't really look like this tree. It has more of these linear 90-degree angles that really differentiate the in and the out groups. Okay? Based on that other sheet that we just looked at with the check boxes, we can create a family tree. So if I gave you similarities and differences between organisms, and you guys did one already in your PGLO, I know that. Okay, based on the genetic similarities and differences, you were actually creating a cladogram. What? Did I say PGLO? Oh, sorry. That's the gene in a jellyfish. Used as a genetic marker in biotechnology. I meant POGO. Sorry. PGLO, POGO, tomato, potato. Because they're different. All right. So tracing phylogeny, looking at fossil evidence first. Okay, and I would say fossils are not my basis of evolutionary evidence. But fossil record is incomplete. We know that, and that's why. It's often difficult to determine the phylogeny of a fossil. Why? We're only using physical morphological structures. We are not using genetics. Now, if I look at homology, this refers to features that stem from a common ancestor. Homo means same, right? So studying the structures that came from the same ancestor, homology. If I look at analogy, that's going to be what? Different analogy. Similar to convergent. Now, we'll talk about that in a minute, but let's finish up with homologous structures. These are related to each other through common descent. Let me talk about that. All wings of birds would be homologous structures, correct? Notice I said birds. Okay? How about the wings of a bird versus the wings of an insect? Do they have a common ancestor? No. So that would be analogy, right? Okay, so this is similarity due to only convergent evolution. The fact that they both had to fly and adapt in an environment that required flight meant that, okay, they have a similar environment that they evolved in, but they do not have a common ancestor. And that would be analogy. Now, analogous structures have the same function in different groups, but do not have a common ancestry. Structures look similar due to adaptation to similar environments, but they will not have that common ancestor. Therefore, we cannot infer homology or homologous structures from that. Brings a whole new meaning to analogies, right? Kind of? No? Yeah. Don't put that on your AP English exam. That won't work. So tracing phylogeny, behavioral traits. We can look at how things behave, right? Parental care, mating calls, etc. Frogs are big on their mating calls, right? Yeah? Spring peepers. And as summer goes on, you get your deep green frogs and your really deep bullfrogs with the ping afterwards. Ping. You ever hear that one? No? We're going to take a frog trip this spring. We'll cross the road. We'll call it Froggert. So, yeah, I mean, what fun is a field trip unless you might, Right? So molecular traits, systematics assumes that two species with similar base pair sequences are assumed to be closely related. So I like systematics. It basically gets rid of all the arguments. That's why I like it. It really do. I don't like to sit there and debate a structure on an organism or the missing link. I just say, let's look at systematics. Let's look at the molecular makeup of given organisms. So two species with different base pair sequences are assumed to be only distantly related. So if I take a look now at a better checklist of systematics, and I look at, let's say, the similarities and differences between different species, so these all, let's say, are a different amino acid, okay? And I said, okay, a cow has cysteine, pig has cysteine. Uh-oh, a horse has tyrosine here. There's one difference, right? And so based on this alone, and be like, oh, geez, it looks like they all have G here. That's the same. So I'd start looking for differences, okay, and I'd be able to count them. Did you guys do this in lab? Yeah. And so the fact that I have a gene chip now, an array that I can actually put any DNA into, and it will spit back to me the actual order of amino acids in that given organism, allows me to quickly tell if things are evolutionarily related. Okay? What? Yeah, you did it in your POGO. Yes. So I know you did this, but I really want you guys to understand that this is the modern way that scientists are talking about evolution. Very few are referring to fossils anymore, okay? Usually we're looking at DNA evidence. So tracing phylogeny, we look at protein comparisons. That's what you were just looking at as an order of amino acids. And then immunological techniques were the degree of cross-reaction used to judge relationships. Trying to see, okay, how are you two related based on the proteins that you make? Which leads into amino acid sequencing, where similar sequence in the same protein indicates a close relationship. That's actually how we know we're closely related to gorillas and chimpanzees and not so closely related to monkeys. Plus, we're missing a tail, right? It's kind of like... I mean, it's one thing to say, hey, we both don't have a tail and we have opposable thumbs. It's another thing to say, hey, look at our systematics and our molecular makeup of our proteins. They're super similar. And that leads to a molecular clock. Do we know the rate at which mutations occur? Yes, for sure. Because of that, if I count the number of differences, I know how long it takes to get those differences. That doesn't mean I know what differences will eventually evolve and occur, but I know that if I have so many differences between me and another organism, I can tell how long ago we diverged, right? Okay. So this assumes a constant rate of mutation over time. And you guys, how do we know this is true, or how can we back it up with science, I should say? True is always able to be disproven later. Truths are always disproven in the future. But let's just think about, what if my molecular clock matched the fossil record in relative and absolute dating within a few thousand years? Would you say relatively accurate in depicting the timeline of Earth? And in fact, the molecular clock falls within a few thousand degrees of accuracy as far as years. And we're talking about a blink of an eye, not even, in the history of Earth when we say 1,000 years, right? And so the fact that these match up with what the evidence we already had in the fossil record and using relative and absolute dating shows us that this molecular clock holds very true, which is very cool when you think about it. All right, guys. So that's the end of this chapter.